So you have heard a couple of presentations in this symposium that are already useful for you to understand what is individualized treatment. But my presentation will in somehow complement the previous two presentations. So you heard that individualized treatment is possible, but it's very difficult. It's very difficult because I liked a lot one slide of Michael Davidson, how many things you should keep in mind when deciding the treatment of our patient. A lot. Bipolar, which phase, depression or mania, mixed, which is the story, which is the predominant polarity, which is the tolerability of the patient, which are previous treatment, were they effective or not effective. So you see how many things you should keep in mind before making a prescription. A lot, probably too many. Probably we are not considering everything when we are in that minute that we have to prescribe a treatment. So only speaking from a clinical point of view, things are very complicated. And probably not all of us are doing what would be possible when we have to consider all the possibilities for the best individualized treatment for the patient. Clinical features are a lot and complicated by themselves, but there is genetic. As you heard from Michael Davidson and before, there are already some findings, but they're difficult to interpret, difficult to use. And once we will have also the genetic finding, it will be even more complicated. So probably our mind would not be sufficient, and we will need some external support. What I'm telling you today is that this external support already exists. There are some devices, software, that help you in finding the best possible treatment for the patient, also including the genetic point of view. What the genetic point of view can tell you that is not so easy to understand from clinical aspects. Well, genetic point of view can tell you something that happens in the brain of the patient. Genetic point of view can tell you what are the specific pathways in the specific patient that are more or less sensitive to specific treatment. Just to make a simple example, you could have a serotonin system that is not working properly in your patient. And so there is no point on prescribing serotonin drugs. Or you can have some specific system in the brain of your patient that is more sensitive to some side effects, or very serious side effects. So this possibility is what we are having partially now, and we will have much more in the future. The possibility to guide our prescription, but to guide not in a specific way. This was the mistake of the paper that Dina showed you of this gathered finding. It's not possible that one single SNP, one single gene can control everything. This is not realistic. This is not a possibility. There is a combination. It's a profile. And with a profile, you can have a big help from genetics. So, just to go back to the data that I will show you today, it is possible, from a genetic point of view, to have a specific profile that gives you a percentage, that gives you a possibility for a better targeted prescription. So, genetic will tell you, okay, you have a bipolar patient, rapid cycling, so from a clinical point of view, you should prescribe valproate. But from a genetic point of view, you have some different indications, so consider also these possibilities. This is what genetic is offering. But how can genetic give this information? Today I will present you one of the latest findings that we are working on to show how a genetic study can be performed. Genetic is of course important in bipolar disorder because a large part of bipolar disorder is due to genetic. So genetic is changing the brain of our patients. Our bipolar patients have something in in their brain that is genetically controlled that makes them bipolar. What is exactly, we still don't know, but we know that some areas are compromised. Hippocampus, some area of the frontal cortex, some connections. And also the response to the drug and the tolerability of the drug is controlled by genetics. This is why genetics is important in all these aspects. And this is why genetics has been studied a lot in the last 15 or 20 years in terms of lithium efficacy and lithium response. Some genes have been investigated, mainly the serotonin transporter. You already heard about it, BDNF. Some specific genes have been studied. And there are some signals, but still we don't have a clear picture. This is why studies are going on. 3 studies did something similar to what we did, and I will present you. These 3 studies, this is the first of the 3 studies that investigated the expression of the genes and their correlation with the response to lithium. And what they found, I will not go into detail, just to give you an idea, some groups of genes are the most important. Neuronal cell adhesion, cell cycle regulators, signal transduction, transcription factors, and glutamate. Don't be lost into the details, but think that the things that are abnormal and that can control lithium efficacy are in what and how the brain behaves under stimulation, the flexibility, the plasticity. And so cell adhesion or glutamate activity, glutamate is something that is very toxic to the brain. So all these results point to the fact that the bipolar brain is not responding well to lithium when it's not flexible enough to receive the lithium benefit. And in the same direction goes the next finding, is another paper that is recently published, that is focused on the glutamate genes. Glutamate is, of course, you've seen very important because glutamate is the target of ketamine, rapid antidepressant response, but probably not all of you know that glutamate is also responsible for depression, because glutamate toxicity in the hippocampus is something that leads to depression. So it seems that glutamate is some kind of big actor in depression and in antidepressant efficacy, and also in lithium efficacy in bipolar disorder, glutamate. So, glutamate is important, but glutamate pathways are not similar across patients. You can have a patient with a good glutamate system or a patient with a glutamate system that has different behavior. And this is why our treatment can impact better or worse depending on the glutamate profile of the patient. So our study was made to answer this question: which are the genetic factors that can impact on long-term lithium and other mood stabilizers effect? So not only lithium. This is the new part of our study. We studied all the possible drugs that can control lithium and other mood stabilizer efficacy. So the sample size is the STEP-BD, is an American multi-center study, very large. The original sample size is about 4,000 patients. And those patients are followed up in a naturalistic way, so reflecting normal functioning of lithium clinics and bipolar clinics. Our aim was to study all the genome to see which were the genes that were associated with long-term efficacy, long-term more than 6 months, of all the possible treatments. And this is the new part compared to the previous studies, that they focused only on lithium and in different follow-up strategy. We focused on all treatments. Focusing on all treatments, we will see this at the end, can also complicate things, because focusing on all treatments, you don't know if the gene that you find are specific for some drug, or if the gene that you find that control for outcome are linked to the disease itself. So, but this, we will answer to this question later. For now, focus on the fact that we study a large sample of bipolar patients treated naturalistically, and we will try to see which are the genes that are influential in the outcome of this sample. Outcome that is considered as the total number of episodes that the patients report during follow-up. And this is the primary aim. As secondary aim, we focus only on depression, and another secondary aim is the focus on mania or hypomanic relapse. These are some genetic details that I know that you are not familiar with this, but they basically serve to be sure that the genetic data that we have are reliable. It means in terms of quality, in terms of people that have a good genetic analysis and so on. I will not go into the detail of course. And the second step, and this is also new that we did compared to other previous studies, we did not consider a single gene or a single polymorphism. We considered a pathway. This is why pathways are much more important than single genes. It is unlikely that a single gene variant controls long-term bipolar outcome. It is much more likely that a system could be a system related to cell adhesion, a system related to learning, glutamate systems. A system can much more reliably influence long-term outcome of bipolar disorder. This is why we also focused on pathways. And just to give you an idea, which were the patients, there were, the sample was about 700 patients, standard criteria, as you see, many previous episodes, many previous depressive and manic episodes, follow-up of about 2 years, so it's sufficient length to have an idea, many visits, standard treatment with monotherapy and polytherapy as we do in clinical practice. As I told you at the beginning, the new part of this study that we consider all possible mood stabilizers, not only lithium, but also valproate and all the other possible mood stabilizers that have been used, and also some of the depressant treated patients. This is a result. I know that for you it could be difficult to understand what this result is. I will briefly explain. All these dots that you see are all the variants within the genes. More than 500,000. We studied more than 500,000 variants for each patient. And the level, the height, is which variant is relevant for the long-term outcome. So, we saw a number of variants, these ones. I know that those names tell nothing to you, but I will explain to you later. We found some of the variants that are very strongly related with bipolar long-term outcome, independently from treatment. And the interesting thing is that those variants, that some people could say are chance variants, are very relevant because they are somehow a story of previous evidence, a story of relevance in bipolar outcome of disorder. So one could say, if we found 5 variants out of 500,000, they could be chance finding, but it is very difficult that they are chance finding if they are already known to be relevant to bipolar disorder. I will show you later why they are relevant in bipolar disorder. Same story for depressive recurrence or manic recurrence. And this is the second result about the pathways, because I will describe you later what are the genes that we find associated with long-term bipolar outcome. And now I'm showing you which are the pathways. Pathways means combination of genes. significant is a pathway that is linked to learning and memory, and is composed by some glutamatergic genes and some other post-transcriptional controls. What is learning and memory pathway? It's a very relevant pathway. Consider our patients from a clinical point of view. Bipolar patients have to learn a different way of facing the disease. The most common problem with bipolar patients is that they do not respond to treatment because they stick on their cognitive style in terms of depression, in terms of hypomania, mania of course even more. And so, of course, one of the big difficulties of these patients is to change their cognitive attitude. Of course, lithium and other mood stabilizers influence the pathways in the brain related to learning and memory. And so, if you have a patient that is more or less flexible in terms of learning and in memory capability, it's obvious that in terms of response will be different. So this is just one example. Cognition is another very relevant pathway. And for depression recurrence, another pathway that we will see a little bit later, it is a regulation of the MAP cascade that I will show you later how relevant it is. This is another even more complicated for you to understand system to look at our results, and I will explain you a little bit now. That is very innovative. It has been created in our group, and we already published in some of the papers. That is even more large, not single SNP, not single gene, not even single pathway significance, but the combination of different genes, also including different pathways, when they are related one to another. And what we look at? We look at the number of SNPs within every gene that are significant, and then we create a global threshold. I understand that this could be complicated, but what is the idea behind this new method? That we are looking at the brain as much as possible as a whole, not sliced into genes or SNPs, as a whole. And so we can understand when a pathway is really influencing something or not. In fact, this approach has been accepted from a scientific literature, and we think that it could lead a little bit more closer to clinical application. Because we cannot use single SNP for clinical application, probably we cannot use single genes for clinical application. We will have to use a combination of balancing in the pathways of the brain for clinical use. And this is obvious, because you can have one gene that is not working in the brain of your patient, but then another one can complement it. So you have to look at the whole chain of functioning of one system in the brain of our patient. And this is the pathway and the enlarged pathways of learning and memory. And the black genes are the genes that give signal of association with outcome. So, again, not going to detail, it means that when a patient has a combination on all these genes in the same learning and memory pathway, it means that this patient will have a learning and memory impairment, less flexible, and in this patient, mood stabilizers will work less. Same for the regulation of the MAP cascade that I showed you for depressive recurrence. MAP cascade is a system that is extremely important, both in antidepressant response and mood stabilizer response, because it contains many genes of the glutamate system, including this gene of the glutamate system that was already associated with outcome in one of the previous studies I showed at the beginning. So you see, it is very unlikely that those are chance finding, because always the same genes pop out, always the same, more or less the same. So it's very difficult to do such chance finding, because out of 500,000 SNPs, if there were a chance, you can have one gene one time, one gene another time. But here, always the same signals come out. So probably these areas are very important. So the last 3 slides to summarize what we have seen. We are talking about predisposition, liability for good or bad long-term bipolar outcomes. So this is an extremely important point, because if we can tell since the beginning for a patient if there is a good or bad long-term outcome in terms of relapses with standard treatment, we can focus a much stronger treatment on the patient that need more it, and don't force it on patients that have anyhow a good outcome. So you can understand from a clinical point of view how important would it be to know in advance. Which were the signals more relevant? In some genes, this, for example, was the strongest that has been already associated with schizophrenia previously, and it changes RNA function. Those other couple of genes have been already associated with bipolar disorder, and these are very interesting, and they have been associated also with antidepressant and antipsychotic response, the same genes. So you see how unlikely it is to they are false positives, because they have already a history of association with response. This also is extremely interesting because it's in the GSK3 beta system. What is the GSK3 beta system? It's the key of lithium efficacy, for example. You know, probably some of you are familiar with this picture. This is how the signal is transducted in specific receptor subtypes, and GSK3 beta is a key factor for lithium efficacy. In fact, from a clinical point of view, some people used also inositol to give to patients, inositol with a spoon, because inositol is probably linked to lithium efficacy. And this gene is just here, is controlling the transduction of the signal of lithium efficacy. So this further encourages us to understand that if you focus on the pathways that are relevant, you can find something that can be useful. And this other genes is also very important because it's linked with the glutamate transduction, this protein here that is in between the chain of transduction of glutamate. So glutamate, as you know, ketamine relevance and toxicity relevance. The other 3 genes, as we discussed, are linked to plasticity, memory and learning, things I told you before, and some potassium channels that have been also associated with bipolar imbalances. The last genes are in the adhesion molecules. What they do? They do the synapses growth. When a patient changes and improves on depression, you have a change in synapses modeling, and so that this system can control the synaptic modeling of our patient. And glutamate receptor, as I told you before. Of course, any study is full of limitation. I wanted to go a little bit into detail for you, that I know that you're not familiar with genetics, to show you how a genetic study is performed and which are the results that come out from genetic studies and how can be useful for clinical practice. Of course, there are limitations. To consider all treatment together can be considered a limitation, but we think that to consider all treatment together is useful, because in practice you don't use only one treatment. Usually you use a combination of treatment. You sometimes have pure lithium, pure valproate, but most often you have a combination of treatment. And of course there are other limitations to that. Technical, I will not go into detail. So the last slide is that it is possible to identify genetic predictors of long-term bipolar outcome. Previous literature and our present study are focusing and converging on some systems. Systems of glutamate, that glutamate is very relevant for depression and antidepressant efficacy, focusing on system of learning and memory that are probably very relevant. And once we will get more exactly in this component of these pathways, we can use it. Michael Davidson person told you that already some companies are offering these services. It probably is too early. Probably results are not very stable yet. But something is there. So I'm quite optimistic that in the near future we will be able to have some contribution from genetics. Not just a prediction, this patient will be a wonderful responder, this other patient will be a very poor responder. No, a percentage of contribution that could be combined with our knowledge contribution. So, these are the results. Also, my slides are here available for anyone who wants or can write me. And the final word for thank you, my people in Bologna that are helping me to work on this very difficult issue. Thank you for attention.
Thank you very much, Alessandro, for this excellent talk. Any questions, comments? Professor Nathan, please.
Thank you. This is a very good session, actually. It's by the end of the day. Alessandro, one thing we are now facing is that clearly some of these pharmacogenetic tests provide some information, but it's not totally clear to what extent this information taken together, it does help to save, let's say, time at hospital or the money we spend on patients. So there are some studies going on. The problem nowadays is the cost of this, but this is probably going to be lower, right? What would be the cost, in your opinion, that would be really accurate, or and whether this is really a reality nowadays?
Okay, so let's start from the second point and then we'll go to the first. The costs are now to get a panel of genes that are relevant around hundreds of euros, so 100 to 300. If you want a complete, exhaustive, complete genome sequencing, now is 10,000, so it's too much. But it will go down. It has been going down since the last 20 years. It will go down to less than 100, for sure, in some years. So it is feasible, it is cheap. Let's go to the first question, what you have in your hand. I like a lot the presentation of one company, that I'm not talking to, not make advertisement to this company, that makes a very simple thing, because clinicians need simple things. Green, yellow, and red boxes. So it means that for your patient, you have green for this list of drugs. It means, okay, they work for your patient and they are tolerated. You have yellow, some problems with these drugs, and you have red. Be careful. Those drugs can give problems to your patient. Very simple. So I think this kind of communication between genetic and clinical practice is very promising. So don't worry, all the hard work, we do it. We don't give you glutamate receptor result or learning memory result. We give you just green, yellow, and red. So be careful, not avoid, but just be careful with the red ones, because they can be less tolerated or probably not working very well with your patient. And then green, you are more easy. So this is the contribution that is already happening. This company is taking a vocal swab, you mail them, and 2 days later you have this green, yellow, and red answer. So it's very easy, very cheap, $200, $300.
Question to Professor Rogers first.
Thank you, Alessandro. Very nice presentation. I am familiar with this simple system that you described, the neuropharmaceutical system. I agree with you that can be helpful. But my doubt when I see this presentation, even before, is always the same. We are using treatment that are unspecific. And the diagnoses are unspecific. Many of these genes can be utilized for many of our so-called illness, schizophrenia, or psychosis, or bipolar disorder. So at that moment, the even neuropharmacy, the simplest system, works in predicting the side effects, the tolerability, but it's less reliable in predicting response or probability of response, because what these genes predict is the probability of response, and in my view are not more effective than using what we know at clinical level. Symptoms, previous history, family history, how is the father, temperament, such kind of things that we use in clinical practice. We are not able to add anything really significant in terms of predicting response or outcome, especially if probably we can have some help in predicting the prognosis, but we are not sure that this is related to treatment or to the nature of the illness. So this is still today the measure. I am with you that in the future probably, but we have to change our way to classify our illness, because this system is finished. Even for geneticists and for regular genetics, in my view, but probably I'm pessimistic and skeptical, as always say.
So to the last point, of course I agree that the nosographic system is very old now, so I think should be changed. About the other points, of course, you have always to consider the clinical aspects. What I suggest, and what everybody suggests, not to rely on genetic findings only. You have always to consider all the clinical aspects, but the genetic ones give you some more suggestion. More aware, more in the direction of tolerability, for sure, because tolerability is much easier. Some of these genes are approved by FDA, like carbamazepine, skin reaction, so tolerability issues are much easier. Outcome is much more difficult. I'm always mentioning one of my patients in my center, that my residents came to me telling, oh, this woman is not improving. I think she's resistant to depression. I said, well, what we have done? Oh, I tried the sertraline, 200, no response. Venlafaxine, 375, no response. What should I do? She's resistant depression. I asked her, but tell me more. What? Well, actually, she lost her husband 1 year ago, and she lost also the job, so it's an economical problem. Now, her son has some serious disease, and she also had some chronic serious kidney disease. They say, okay, how can this person be a responder to venlafaxine? We have other external issues. So I understand that this is very complicated. It's just a small help that I think this green and yellow and red is a good point.
You know, we take one short question because we have to go to the next.
Don't you think that on the black spots you have, you should use statistics which look at this pattern in the raw data? This would boost tremendously the associations. That's for clear. It's very cheap. The second thing is, don't you think that the pathways have to be looked at for all the cells of the immune system more than any other, or the neural cell? I think that's the way forward, because then you see what they're really doing in the body. Yeah.
Thank you for the suggestions. Thank you.
Thank you very much. And now I would like to introduce Professor Kainina, who's professor at the Indiana College and also associate professor at the University of Minnesota.